Hello students. So in this video we will continue this is a complex tissues. In the previous video we discussed about xylem, the water conducting tissue. And now let us jump into the next complex tissue, that is phloem, which is a food conducting tissue. And this phloem is also known as leptome or bast. Leptome or bast are the alternate names of phloem tissue. And what is the function of this phloem? Of course, everyone knows it is a conducting tissue and it conducts the food products, especially the food will move in the form of sucrose solution. Sucrose solution from source to sinks. What is source? Source is the production centers. Sinks are consumption centers. And of course, we know that the food is prepared by the green parts of the plants. What are those green parts? Leaves. From leaves to all the parts which cannot prepare their own food material, like what roots, apical buds, axillary buds, flowers, fruits. These are all what they won't synthesize the food material, but they require food. And to those parts, the phloem will translocate these food materials. So these parts which I have listed out, they are all what sinks, and the leaf is what source, production center, and the consumption centers are these roots, and other parts. So, but remember one thing, that one difference will be there. While discussing about the xylem, we have come to know that xylem is conducting water and minerals, which is called sap, in upward direction. Means what? Only one direction. But the phloem can conduct in all directions, and especially we can say bidirectional flow will be there. Means what? As I said, the leaf is the source, and the root is the sink. Okay, so now the food material, when it is translocated continuously, the food may not be consumed by the root. Sometimes what will happen, the roots will store these food materials. And during certain conditions, environmental conditions, the shoot system may not be able to synthesize the food material. Then how the plant will survive? So now from the root, the food material which is stored will come back to the shoot system. Come back to the shoot system through which tissue? Phloem only. Earlier, through which tissue it went into the roots? Through phloem only. Means what? This is what you have to understand. So this is what bidirectional flow. So that is the main difference between the xylem and the phloem. While xylem is conducting the water and minerals in one direction, that is unidirectional flow, the phloem will show what bidirectional flow. Okay, and now let us say this phloem we are talking under the title complex tissues. Complex tissues means what? Group of dissimilar cells. As we saw, the xylem is composed of different types of cells. Even the phloem is also composed of different types of cells. So what are those cells? Sieve elements, companion cells, phloem parenchyma, phloem fibers are bast fibers. So sieve elements are of two types, sieve cells and sieve tubes. And you have to remember these. Sieve cells are present in pteridophytes and gymnosperms. Sieve tubes are present in flowering plants, that is what angiosperms. So I hope you're understanding. Sieve cells are present only in pteridophytes and gymnosperms, and sieve tubes are present in angiosperms, and the sieve tubes are absent in pteridophytes and gymnosperms. So, presence of sieve tube is a characteristic feature of angiosperms. We said the vessels of the xylem, presence of vessels is also a characteristic feature of angiosperm, like that sieve tubes are present. And the sieve tubes are always associated with companion cells. Okay, so these are also present only in angiosperms. And actually, the main food conducting cells are what? The sieve elements and the companion cells. Okay, and the phloem parenchyma, of course, it will conduct the food material in the sideward direction. Okay, that is called ray parenchyma. Okay, so that we will see later. And now remember one thing, that among all these cells, the sieve elements are living cells, and when they are young, means what I am talking about, both sieve cells and sieve tubes. When they are young, they are with nucleus. So that means they are uninucleated cells. But on maturity, once they become matured, so the matured sieve cells and sieve tubes will become enucleated. Means what? The nucleus will disintegrate, or nucleus will degenerate. So they are without nucleus, enucleated cells. So that's why don't forget that the matured sieve cells and matured sieve tubes are enucleated, but young sieve cells and young sieve tubes are nucleated. Okay, but that means they will contain the cytoplasm as it is. So that's why they are living cells. The sieve tube, sieve elements are living. That means both sieve cells and sieve tubes are living. Companion cells are living. Phloem parenchyma is living. And phloem fibers are dead. So the majority of the cells associated with the phloem are living cells, and few of them are dead. So that's why the phloem is described as living food conducting tissue. Of course, we discussed while talking about xylem, that is dead water conducting tissue, because majority of the cells associated with the xylem are dead cells, but majority of the cells associated with the phloem are living cells. Okay, now let us say the details of these sieve cells. Okay, and don't forget the sieve cells are present only in the pteridophytes and gymnosperms, and the sieve cells are elongated cells, spindle shaped cells, and they are with only primary walls. Just remember that these living cells are always with the primary cell wall, which is made up of cellulose. Okay, cellulosic walls, and having tapering ends, and you can see here they are connected at their ends. But here you can notice one thing, that is some the lateral walls, means what, the side walls are composed of some pores, and these pores are called what? Sieve pores. Means what, as they look like the pores of a sieve. So that's why they are called what? Sieve pores. But you can see the sieve pores are arranged in groups, and this each group of the sieve pore, these groups, is called what? Sieve areas. Okay. So due to presence of these sieve pores in the form of sieve areas, I hope you are not getting confused. So the pores are called sieve pores, and the group of sieve pores is called sieve area. So as the cells are provided with sieve areas, they are called what? Sieve cells. And these are associated with what? Pteridophytes and gymnosperms, and they conduct the food material through them, and they contain what? Cytoplasm, and also nucleus when they are young, and when they mature, the nucleus will degenerate. So these are what? Sieve cells. And of course, so you can see the movement will be like this. And remember that once their nucleus degenerate, and who will control the metabolic activities of these cells? For that purpose, some parenchyma cells are modified, and these modified parenchyma cells are called albuminous cells. And you can see these albuminous cells are present by the side of these sieve cells, and they are connected through plasmodesmata. Plasmodesmata. So, sieve cells and albuminous cells are present in pteridophytes and gymnosperms. Sieve cells plus albuminous cells are present in pteridophytes and gymnosperms. Now, let us say these sieve tube cells. You can see here the sieve tube cells are elongated tubular cells. Elongated tubular cells. And I'm not showing the lateral walls, but lateral walls are not very important. And here you will find one special feature that they have what? They are arranged one above the other in a row. So that's why you can't separate the cell here. The cell and the cell, they are all connected to each other. And you will find what the cross walls here. And these cross walls are generally we call them about septa. And the septa are having some special feature you can see here. So you will find what sieve pores here. Sieve pores. And that is what specialization here. The sieve pores are not that means not concentrated on the lateral walls, but they are concentrated in the sieve, okay, in these cross walls are septa, and that's why this cross wall or the septum with sieve pores is called sieve plate. I hope you are understanding. You see, sieve plates are associated with sieve tubes, not with the sieve cells. Okay, and through these sieve pores of the sieve plate, the cytoplasmic strands will move from one cell to other cell. So they're all connected. And one more thing, the cytoplasm will have some fibrils which are made up of P protein fibrils. P protein, that is phloem protein. Protein fibrils are there. And one more thing, you will find some amount of a material, okay, which is soluble material, and this material is called callose material. And this callose material, okay, will accumulate during winter season. And whenever it accumulates during winter season on the sieve plate, so it blocks the sieve pores, and because of which this transport will be blocked. And during other seasons, this callose will dissolve, and as a result of which the pores will get open, and that's why the transport will take place. So like that, the callose material is okay, regulating the movement of the food materials through these sieve tubes. But as I told you already, so these cells, when they are young, will contain nucleus, and whenever they are matured, they become what? Enucleate. So, but who will control the metabolic activities? So, for that, you will find what some cells associated with it, and which are called companion cells. Okay, companion cells are always associated with sieve tube cells, and they are arranged by the side of these cells, and they are connected through plasmodesmata. So these connections are plasmodesmata, and you can see the nucleus of the companion cell. So the nucleus of companion cell will control the activities of both companion as well as sieve tube cell. And one more thing is that it regulates the pressure in these sieve tube cells. Regulates the pressure gradient in the sieve tube cells. Okay, and one more thing we have to see, and these companion cells and the sieve tube cells, companion cells. Okay, let me write like this: sieve tube cell and companion cells originate from one mother cell or same mother cell. So that's why these two are called sister cells. Sister cells. And you know that what will happen to the sieve tube cell when the companion cell die? Even the sieve tube cell will die. Okay, so that's how they are involved, or that's how they are related. So that's why they are called what sister cells. Okay, and okay, and now we can see the parenchyma, which is present here, is called what? Phloem parenchyma. Phloem parenchyma. And these phloem parenchyma cells are also connected to these sieve tube cells through plasmodesmata. And they are living cells, thin walled cells, and they have cytoplasm and nucleus, everything. And what is the function of this parenchyma? Storage of food materials. What was this parenchyma? When it is present in the okay, in the secondary phloem, okay, then you will find its arrangement. Okay, there you will find the two types of parenchyma based on the orientation. When they are arranged like this, parallel to other cells, so it is called what? Axial parenchyma. And when they are arranged perpendicular to these cells, okay, so like this, if they are arranged like this, so these are called ray parenchyma. Ray parenchyma. And that ray parenchyma will help in what? Radial conduction of food materials. So don't get confused. We discussed about this ray parenchyma even in the xylem. So that ray parenchyma of the xylem helping in the radial conduction of water. But this ray parenchyma is helping in the radial conduction of food material. Means what? Sideward conduction of food material. So like that, you will find this parenchyma. And one point you just remember: this phloem parenchyma is absent in monocots. Phloem parenchyma is absent in monocots. Okay, and now the phloem fibers, which are sclerenchymatous fibers, and these fibers are with lignified walls, and they provide what? Strength to the phloem, but they are generally associated with secondary phloem. Okay, there are very few in the primary phloem, but they are more in number with secondary phloem, and that's why they are called what? Bast fibers. And these bast fibers are, of course, they provide strength to the phloem, but we make use of them. That's why most of the fibers that are used by us are called commercial fibers. Okay, for example, the jute fibers. Okay, these hemp fibers. Okay, flax fibers. Okay, rami fibers. These are all commercially used in textile industry, and also some of them are used in making gunny bags. And all these fibers are okay, are present associated with secondary phloem. Secondary phloem fibers, so which are called as bast fibers. Okay, and just remember as a point, these ramic fibers are the longest fibers. Ramic fibers are longest fibers. And the flax fibers are pure cellulosic fibers. Pure cellulosic fibers. Okay, so and of course remember one thing that among these different types of cells, what are associated with the angiosperms? Don't get confused. So in angiosperms, the phloem is composed of sieve tubes plus companion cells plus phloem parenchyma plus phloem fibers. Okay, so this is what you will find in dicots. And when we come to the monocots, okay, let us see the elements of phloem in monocots: sieve tube cells, companion cells, phloem fibers. These are present in monocots. And what about these pteridophytes and gymnosperms? Sieve cells, albuminous cells. And they may have phloem parenchyma and also phloem fibers. So, among all these cells of the phloem, which are involved in the conduction of food material? The sieve elements, companion cells, phloem parenchyma. And which cells are involved in providing the strength? Phloem fibers. Okay. So before concluding the lesson, let us see some more important aspects here, like what the vascular tissues, the xylem and phloem. They're classified based on origin and based on differentiation. Based on origin, when the xylem and phloem, when they are derived from the procambium of apical meristem, when they are derived from the procambium of apical meristem, then they're called primary xylem, primary primary xylem, and primary phloem. I hope you are understanding this point. So the initial growth of the plant is called primary growth, and the primary growth is contributed by the activity of the apical meristems, and the apical meristems will have different zones, like what protoderm, peribulum and pleroma. And the pleroma can also be called as procambium here, and that procambium will produce the vascular tissues. So the vascular tissues which are originating from this procambium are called primary vascular tissues. Okay, and what are those primary vascular tissues? Very simple: primary xylem and primary fluid. And at the time of secondary growth, one cambium is formed, that is called vascular cambium. And the vascular cambium also produces vascular tissues. Which vascular tissues? Very simple: secondary vascular tissues. So just remember, you should have this clarity. The procambium will produce primary vascular tissues, whereas vascular cambium will produce secondary vascular tissues. And so, as you called the primary vascular tissues as primary xylem and primary phloem, in the similar way, as vascular cambium is producing secondary vascular tissues, they are called as secondary xylem and secondary phloem. But remember one that there is certain differentiation or difference you will find between this primary xylem and primary phloem. Here, based on differentiation, the primary xylem is called, the first formed one is called protoxylem, and the later formed one is called metaxylem. So the first formed ones are small in size, and they will have annular and spiral thickenings. And the metaxylem elements are large in size, and they will have scalariform, reticulate, pitted thickenings. So in that way, we can differentiate protoxylem and metaxylem, and this is applicable to only to primary xylem, not to the secondary xylem, because the secondary xylem is produced continuously in the form of rings, and which we call them as annual rings. So even the primary phloem size variation will be there, and we should not expect the thickenings, because these are living cells. So the smaller ones which are formed first in the phloem are called protophlloem, and the later formed ones are called metaphlloem. So even in the phloem also, we won't find this differentiation during secondary growth. Okay, so this is what you have to remember, these points at the end. Okay.